It was good to see you actually and do this interview regarding the small delays in time. I know it's close to Christmas time, but I'm grateful that you took the time for me and my research and you find the
Absolutely.
work interesting.
Yeah. No, Mark, I'm excited to talk to you. I was hoping you could introduce yourself and provide a general overview of your research.
Sure. That would be nice. I am Mark Dennis Retrato. I came from the Philippines, but now I'm doing my PhD here in Uppsala University in Sweden. I already have a defense date, so I'm finishing fairly soon after the holidays. And what I am into is on method development for reinforcing or analyzing the stability of parenteral nutrition products of a pharmaceutical company because I am technically hired as an industrial PhD student.
As you may have noted, I'm interviewing people who are doing chromatography, mass
Yes.
spec. I haven't interviewed anyone yet who's doing UPLC-MS. Could you discuss why that method was the best approach for you?
That's a good question. For me, myself, I actually not just do UPLC, but from the poster that you have an interest in, it was UPLC. Actually a separation based or chromatography based mass spectrometrist also as well. So I did some work on GCMS before. I also currently doing work in a supercritical fluid chromatography as well. This particular study that that we would like to talk about now is I've done it using PLC in tandem with the mass spec. So why? Because this particular project, it involves screening and analyzing. It's technically a targeted phospholipid kind of work, a targeted lipidomic kind of work, featuring phospholipids and other components in a pharmaceutical raw ingredient that is used for total parenteral nutrition. So we wanted to investigate on how phospholipids can be quantified easily and routinely because we wanted to do a technical transfer after this. Because I just want to stress out that even if I'm doing an academic work, the main goal is to actually do an academic work that is reproducible outside. Therefore, we wanted to have a method that is fairly simple, that is doable, and actually pliable for method transfer. And for these types of analytes, we investigated it before using GC. GC can work, but you need a lot of derivatization reagents for that in order to convert this to become volatile analytes, which is quite hard because there are such huge molecules. We can also apply other types of chromatography, like supercritical fluid chromatography, but we are leaning towards UPLC MS type of instrumentation because we thought that this was the best approach for our type of analysis. And not only that, for the type of sample that we have, because we wanted to do analysis that you don't need to go through a lot of sample preparation. We can just do pre-concentration and then evaporation. Then we can do 2 to 3 just elementary steps and it hasn't have to be so complicated in order to do so.
Very good. I appreciate you explaining that. What criteria did you use to select the mobile phases and column for the UPLC setup?
That is a good question. But since I'm doing method development, but first we can also go to the mass spec first because for the mass spec, we wanted to choose a detector that can actually encompass the key phospholipid or key lipids or the target compounds that we wanted to analyze. So I did direct infusion first with the MS and checked whether I have interferences with that. Then afterwards, after the direct infusion studies in the mass spec, that's when I did the method development using the column and stationary phases. For the column, we had a generic screening with some of our generic mobile phases A and B. And then after that, you just change the gradient all throughout. if your analysis is reproducible enough, then that's when we try to consider, oh, this mobile phase must be good, or this mobile phase B must be good. But in our actual method itself, we planned and we executed that the first mobile phase should be polar enough because phospholipids are mostly polar and we want to elute all of them on the first stage of the chromatography. And later on, the huge ones that is difficult to be removed by the mobile phase that we have or the mobile phase that we assigned for mobile phase A. So that's how we were able to achieve the separation and the detection that we have using that method.
Awesome. I know you touched on this
Mhm.
for a moment, but how else do you ensure the accuracy and the precision of your method during the validation step?
I think I have touched it a bit, but I would like to expand on that. Since the interest of this study is a pharmaceutical product or a raw material for a pharmaceutical product, rather. Therefore, there are a lot of compliances needed to be addressed. And we are using, are you familiar with the ICH Q2R2 guideline? It's the
No,
international.
I'm not.
Okay. Then I can explain further a bit. It's the International Council for Harmonization, Q2R2. So it's a document. So it's a document that actually states how much of an analytical method must be compliant in order to say that it's a validated method and to be assessed that it is a reproducible or an accurate method either. So it's accuracy and precision in per se. And these ICHQ2 R2 guidelines, they are actually the basis of the US FDA, the Food and Drug Administration, or here in Europe, the EMA, the European Medicines Agency guidelines, on how to establish analytical procedures when you are studying a pharmaceutical component or an API or an active pharmaceutical ingredient, for instance. We have this document, but of course, the samples change from time to time. The document itself is subject to some interpretations, and therefore we need to assess ourselves on how we're gonna do this. So therefore, we establish some parameters like building a system suitability study that is technically an injection in the chromatography that tests the feasibility of the method just before running everything else. We also did some matrix matching. When you say matrix matching, we wanted to construct, for instance, a calibration curve, which is based on linear regression. These calibration curves must be matrix matched. It's supposed to have the same nature of the component of the pharmaceutical component we are analyzing. It should be closely related to the one that we are using for the calibration itself. Because most analysis, they use solvent calibration, but we are doing matrix match calibration for it to match the actual material with or without the analyte or with or without the analyte of interest. But that's also a different story because in matrix matching, you also need to do some sample pre-treatment for you to say that, oh, it's really matrix match. And of course, this since this is a targeted lipidomics project, so we are using also internal standards which are deuterated for the quantification to become more reliable and structurally related also at the same time. But in reality, of course, it's really expensive to have one internal standard per each compound. So we actually have a standardized internal standard that caters one, for instance, for a group of analytes that are feasible due to matrix matching that we did during the calibration process. And also establishing some quality control samples, one below the limit of detection, one in between the calibration curve, and one way above the calibration curve. So these things are considered security measures on how we can ensure the accuracy and precision of our method during the validation. apart from the stability studies that we are making, because the main point of the project is not just the accuracy and precision of the numbers that we generate, but on how to actually gauge the stability of the product or of the raw material once it is incorporated with the pharmaceutical formulation.
Very good. Could you discuss any unexpected findings occurred during your analysis?
Sure. Quite many, but I can just discuss some of it. For the unexpected findings, for the phospholipids, especially phosphatidylcholine, since the product is basically egg-based or egg yolk-based, so there's no secret to that. So it's mostly composed of PC or phosphatidylcholines. Sometimes we're only expecting like maybe 2 or 3 phosphatidylcholine species, but apparently we have a lot more. For instance, if you have the poster, for instance, as a reference, there is one analyte that is annotated as PC384 or 364, if I'm not mistaken, that has 4 peaks. And usually we just expect it to have a 2, maybe co-eluting peaks because we're only expecting 2 species. But here we found 4, for instance. And those 4, they are actually corresponding to several possible combinations of the phospholipid that has the condensed formula, for instance. Because to everyone that is not familiar with the phospholipid, so a phospholipid is a lipid with a polar group, a polar phosphate group, and 2 tails, and those 2 tails are mostly fatty acids. And with those combination of fatty acids, because there's a huge range of fatty acids to choose from, so therefore there are a lot of possible combinations that constitute the condensed formula. And therefore, in our findings, we found out that the 2 prominent ones, for instance, for this particular condensed formula, apparently there is 4 that is actually detectable and quantifiable. So these are just one of the examples of our unexpected findings. We also found out that the lysophosphatidylcholine or the LPCs, for instance, their increased ratio in the product has actually an effect towards its stability, for instance. So those things that we're looking more closely into. So those are the unexpected findings from our current work that we wanted to explore more.
What implications do your findings have for the quality control processes of TPN products?
This is a question that I would like to expand when it comes to the applicability of our method. One, since I talked about tech transfer or technical transfer earlier, it is possible to have all of these key lipid species in one method because most of the work that has been done recently are mostly untargeted for this type of work. It's about profiling, but this one, it's a targeted quantification of these key phospholipids or lipid components that can actually be useful not just for our type of sample, but for clinical studies also as well. For instance, if you have a blood, plasma that you want to monitor lipid content, then it can also be possible. It's just that we need to tweak a bit when it comes to sample preparation because for a pharmaceutical product, it's mostly a major component and it's not an excipient or it's not a residue. But of course, if it's clinicals that we're talking about, sometimes they exist in lower amounts. Adjustments are needed to be made, like pre-concentrating or nitrogen evaporation and reconcentration afterwards. So those things. But apart from the possibility of it to be technically transferred and technically simplifying the method, it is also possible to use the method in different stages of pharmaceutical manufacturing or pharmaceutical production. We can use this for raw material assessment. We can also use this when it's compounding or when everything is mixed up. And if you have the final goods or the FG as some people use as a term. So in any stage of the study, the method is actually flexible. to analyze the lipid contents. So that's what I discern as an implication for QC processes in these products.
That makes sense. One question I didn't have drafted, but I was hoping you could touch
Mhm.
on was were there any specific challenges you had in sample preparation that was difficult or didn't go well?
Yes, indeed. Solubility. So solubility is one, and stability is two. So we optimize a lot of solvent combinations for it to work. And we also optimize the extraction method also from the raw material so that it can work also as well. And we also had some issues with some losses. So those are one of the challenges, and also how to track the internal standards. Because our deuterated internal standards, we put it in the start of the extraction. Then, of course, we can assume that once you do sample preparation and you put your internal standard at the beginning of the process, it should be nice and correct. However, when we do that, it's because it's prone to sample loss. One of the losses with internal standard is technically corresponding to the losses of the sample, right? And sometimes it's really hard because we cannot detect the internal standard anymore. But since the sample is a pharmaceutical ingredient, so technically it's in its purest form, so to speak, or the raw material is almost close to pure form. So therefore, it masks the amount of the internal standard present. So it was really hard to quantify at first. So those issues in sample preparation we have encountered before, tried to resolve some and tried to tweak some so that it fits the bill.
Okay. Just out of curiosity, for the nitrogen evaporation step, you mentioned the Eppendorf tubes. I'd be
Yes.
curious to know what the batch size was, how many you were doing at a time, and then also anything you could tell me about the starting volume and the ending volume that you were trying to concentrate to.
That's true. That's right. We usually put 1 milligram of phospholipid in all of the raw material in the F&R tubes. Do some small extractions up to 2 ml. And after the extraction, centrifuge, and whatnot, reconstitution, and put it in the nitrogen evaporate. The final volume before the nitrogen evaporation is around 1. But when, of course, if it's too high, you need a higher rate to have the nitrogen evaporator on, because sometimes we tried it at a lower rate, it was too slow. And of course, it impedes analysis time, especially these phospholipids are somewhat stable, PC for instance, but the rest, because for instance, LPC or phosphatidylcholine is actually a clean phospholipid. We also have some findings that if the analysis preparation time for the samples and the standards are longer, we also find more amounts of the lysophospholipids as compared to a shorter analysis time. So that is also a bit of a compromise when it comes to these things. To answer your question, after the nitrogen evaporation to dryness, we put it 200 microliters of solvent before the analysis.
Awesome. Yeah, and that's one of the main applications for nitrogen evaporation is to do that final step down to dryness.
Yes.
And certainly what you mentioned, sample preparation time is critically important. And that's why having the right tools is
Mhm.
so crucial. So you're not spending excess time
True.
doing the preparation. Finally, in what ways do you think this research could impact patient care?
This is a broader aspect on why we do things, right? For me personally, why I accepted this work before as a PhD student in this particular project, because not that I really want to dwell into pharmaceutical products and in the pharmaceutical industry afterwards, but it's a form of a personal mission or advocacy also as well, because I do have some medical complications such as asthma, to name a few. I would really like to have something that is useful. I really appreciate cutting edge researches that are currently going on with chromatography and mass spec. But I just feel that this hits personally. This hits home. I feel like when this work is done, for instance, the ending of this work is to actually assess stability of pharmaceutical products that are consumed by patients. Critically ill patients, for instance, that cannot ingest food because that's what a total parenteral nutrition product is for. I feel at some point, with my small contribution, I feel like I am part of something that is valuable for patients by doing this technique that is used eventually for quality control, for a routine analysis per se, for checking the quality of these products. Then I'm pretty sure that I made even a small one, a bit of an impact at least to the product that has been made and that has been administered to a patient or two. And I think that's it. That's how can I say it.
This work is super important, Mark, and I was really taken back by your poster and was thrilled to learn more about it today. So I really appreciate you speaking with me.
Thank you, David. Of course. You're welcome. You're welcome.